Hello, AP Bio. Today we're going to wrap up our discussion of protein synthesis by talking a little bit about how proteins end up where they need to be and what happens to a polypeptide chain once it's been made. There are some things that happen after translation to kind of finish off the process of getting a protein ready to do its job. So first, just a quick review. When we learned about cell structure, we talked about ribosomes, and we talked about two different types of ribosomes in a cell. There are some that are called free ribosomes and some that are called bound ribosomes. The difference between these two has to do with the location in the cell and also what type of proteins they make. Free ribosomes are the ones that are just floating within the cytosol in the cell. They're not attached to anything. They produce proteins that are mainly for use right within the cytosol. For example, the enzymes that help with the process of glycolysis that happens in the cytosol, those enzymes would be made by free ribosomes, and then those enzymes are just released into that fluid that fills the cell so that they can carry out that reaction. Bound ribosomes, on the other hand, are the ones that are attached to the rough ER. So bound means attached. They're found on the rough endoplasmic reticulum. Those produce proteins that are either going to be transported to the cell membrane or that are going to be exported and secreted out of the cell. Once they go into the rough ER, they're kind of within that endomembrane system, and they eventually get pinched off into a vesicle and exported from the cell. And so what we're going to talk about first today is how a ribosome knows whether it needs to bond to the rough ER to export a protein out of the cell or whether it should just hang out in the cytosol. Because one thing that you may not have realized is that ribosomes can move from one location to the other. If they're floating free in the cytosol, they can move to attach themselves to the rough ER. And once they're done making that polypeptide, they can also release from the rough ER. And that difference is also coded in DNA. We've been talking for the past couple of days about how DNA codes for messenger RNA, which holds the code for a polypeptide. Well, this diagram demonstrates what happens if that polypeptide is part of a protein that needs to move into the rough ER. So you can see in this diagram, this light blue organelle at the bottom is the rough ER, and the inside of it is labeled the ER lumen. That's the space within the rough ER. The way that this works is if a protein needs to be in the endomembrane system, if it's a protein that's destined for the cell membrane or to be secreted from the cell, the first few amino acids of that polypeptide chain are what's called a signal peptide. It's a specific sequence of amino acids that start to come out of that ribosome. And that sequence of amino acids is recognized by another type of protein called a signal recognition particle that basically just floats around the cell looking for this little green sequence of amino acids. And you can see those two fit together. And so when the signal recognition particle finds this sequence of amino acids, it bonds to it. And the job of that signal recognition particle is to pull the ribosome over and attach it to this receptor protein on the rough ER. So the signal recognition particle notices this little flag that polypeptide is waving saying, hey, I need to go over to the rough ER, and it pulls that whole complex of molecules, even as translation continues happening, and attaches it to the rough ER. So then from that point, the rest of that protein is built into the rough ER. You can see it's kind of being put right into the middle of the rough ER as that happens. Once this polypeptide is finished, the signaling peptide is removed. Those first three amino acids were just a signal to get this ribosome to the rough ER. They're not actually part of the finished polypeptide. So they are cleaved off. They are separated from that polypeptide chain at the end. And then you are left with this protein that is then inside the rough ER. And it then gets transported through the rough ER and eventually through a vesicle to the Golgi apparatus, where it gets packaged up and sent off to wherever it needs to go. The ribosome then, after it's finished making this protein, can detach from the rough ER and continue to float around the cytosol, either to make proteins that need to go into the cytosol as a free ribosome, or if it translates another messenger RNA for a protein that needs to be exported, this process would happen again. So the protein itself, that polypeptide itself, signals that it is a protein that needs to be exported from the cell or to the cell membrane. Once a polypeptide chain has been made, whether it's in the cytosol or in the rough endoplasmic reticulum, there are modifications that need to be made after translation is complete. A polypeptide chain is not a finished protein. Sometimes there are chemical modifications that need to be made to some of those amino acids. Other molecules or molecular groups might need to be added to parts of that polypeptide chain, and that could include sugars or lipids or phosphates or other chemical groups. Sometimes certain amino acids need to be removed from a polypeptide chain, like that signaling sequence that we saw at the beginning of the last slide. We'll look at another example of this one in a minute. The protein, of course, then also needs to fold up. And the process of protein folding also happens with the help of other proteins. But that, like we talked about before, there are several levels. It folds into its secondary structure as the backbones of those amino acids form hydrogen bonds, and then into its tertiary structure as the R groups interact. With some proteins, like this one at the right, this is a picture of a hemoglobin protein. It also has that quaternary structure where you actually have multiple polypeptides that come together to create a functional protein. So hemoglobin is actually coded for by 4 different genes that make 4 different polypeptides. Each of these colors represents a different polypeptide chain, and all 4 of them need to be transcribed and translated, and then those 4 polypeptide chains come together to form the finished hemoglobin. Here's one other example of a protein that you're familiar with that needs some significant modifications before it's finished. Insulin is built as one big long chain. It does actually have a signaling particle that's cut off. That's not shown in this picture. But then in an insulin protein, the entire middle section of the chain also needs to be cut out. These amino acids are not part of the finished insulin. A finished insulin molecule looks like this. It has 2 separate chains, like the green and the orange parts here, that are attached by a few covalent bonds. And then, of course, those chains also curl up. You can see some of those alpha helix shapes, and the finished shape of insulin looks a little bit more like this. So there are many things that need to happen to a protein before it's totally finished and functional, and then it goes out into the cell or gets exported through the cell membrane to do its job. On Thursday, we will be talking a little bit more about this process, but we'll be focusing a little bit more on what happens when this process goes wrong. If there are mutations in the DNA, how can that possibly affect the building of a protein? So that'll be our topic for Thursday. Remember, tomorrow we are having a live assessment, a partner assessment during class, so contact me if you will not be there and you haven't contacted me yet. Your protein synthesis modeling activity is due by tonight, so finish that up and turn it in if that's not already done. And I hope you have a great rest of your day.